When you look around, you can see plants. Do all of them look similar? No. There are trees, herbs, shrubs, climbers, creepers, etc. Some plants look externally similar and some look different. Do you think the internal structure of all plants is similar? No, right? Internal structure also shows some similarities and differences. The study of internal structure of an organism is known as anatomy. In this video, we will learn about the anatomy, that is the internal structure of higher plants. Now tell me, what is the basic unit of a plant? Cell, right? Similar cells which are similar in their shape and size and perform same function, they group together to form tissues and tissues group together to form organs. These organs also differ in their functions and structure. The structure and functions of tissues vary depending upon the location in which they are present in a plant body. So, based upon the structure and location in the plant body, tissue system is divided into three types: epidermal tissue system, vascular tissue system, and ground tissue system. First, let's learn about the epidermal tissue system. You all might be familiar with epidermis, right? What is epidermis? Epidermis is the outer layer of an organism. This epidermal tissue composed of what? Epidermal cells. Here you can see the picture of epidermal cells. These are the epidermal cells. Now, what is the specialty of epidermal cells? You can see they are elongated in structure and they are compactly packed. What do you mean by compactly packed? They are tightly packed. There are no intercellular spaces between the cells. They are compactly packed and they are parenchymatous in nature. We have learnt about parenchymatous tissues in 9th grade. They are storage in nature with small amount of cytoplasm and large nucleus. Epidermal cells are elongated in structure, compactly packed and parenchymatous in nature. A waxy layer is present covering the epidermis, which is known as the cuticle. It is absent in this picture. Let me draw it. So this is a waxy layer covering the epidermis, which is known as cuticle. This waxy layer prevents the excess loss of water. But this cuticle is absent in roots. What could be the reason why cuticle is absent in roots? Roots are meant to absorb water and nutrients from soil, right? If cuticle is present, it may block the water absorption and hence they are absent in roots. An important structure in the epidermis of leaves are known as stomata. We all are familiar with the function of stomata. It regulates the process of transpiration, that is the water loss from leaves, and also helps in gaseous exchange for photosynthesis. Stomata is the plural form and singular form is stoma. Each stoma has two bean-shaped cells. This is one cell and this is another cell, which are bean-shaped, and they are known as the guard cells, which encloses a pore, a hole in between, which is known as the stomatal pore. In grass, the guard cells are in the shape of dumbbells. It could be like this, dumbbells. In grass, the guard cells are in the shape of dumbbells. This allows rapid opening and closing of stomata and will help them to get adapted to different water available environment. For example, if the availability of water is very less in the environment, this dumbbell shaped cells can close the stomatal pore very rapidly to prevent excess water loss. Now the inner wall of guard cell is thick and outer wall is thin. What could be the reason? The thick and rigid inner wall restrict expansions, causing the cell to bend outward when turgid, and the thin and flexible outer wall expands easily, helping the cell to curve, and this curve will open the stomatal pore. The difference, that is the difference between the inner and outer wall enables the guard cells to efficiently open and close the stomatal pore for transpiration and gaseous exchange. Guard cells possess chloroplast as well. You can see these green spots in the guard cells, which are the chloroplast. Sometimes a few epidermal cells near the guard cells become specialized in their shape and size and are known as subsidiary cells. Here, this epidermal cell changes its shape and size, and now this can be called as subsidiary cell. Subsidiary cell. They assist the guard cells during opening and closing of stomata by absorbing or releasing water and ions. The stomatal pore or stomatal aperture, this one, the guard cells and these subsidiary cells, they together called stomatal apparatus. The epidermal cells bear hairs especially in roots, which are single-celled elongation of the epidermal cells. So this is a root, and here you can see the root hairs. These are the single-celled elongation of the epidermal cell. They help in absorption of water and minerals from soil. The epidermal hairs in stem is known as trichomes. Imagine this is a stem, and here also the hairs are present. These hairs are known as trichomes. They are known as trichomes. They are multicellular. So if we take one single stem hair or trichome, it could be like this. There will be multiple layers present. This is a trichome, but in a root, the epidermal cells are single-celled. Trichomes can be branched or unbranched and may be secretory. They secrete oil and resins. For example, in tomato plants, trichomes secrete resins for defense. They help to prevent water loss by transpiration by creating a barrier on the epidermis. These trichomes, actually, they create a barrier on the epidermis to prevent excess water loss. Next is the vascular tissue system. They help in transportation. The xylem and phloem, you all know. Xylem and phloem are transporting tissues. Xylem and phloem together constitute the vascular bundle. In the stems of dicotyledonous plants, a special structure called cambium is present in between the xylem and phloem. This is the xylem and this is the phloem. And there is a special structure. You can see this green line, which is known as the cambium. This special structure, cambium, is present only in dicots. And it is a meristematic tissue. You know that meristematic tissues, they produce new cells, right? So it produces new layers of xylem towards inside and new layers of phloem towards outside. This increases the thickness of the stem and root. The vascular bundles of dicot which have cambium present in between the xylem and phloem are called open vascular bundle. In monocots, the cambium is absent in between the xylem and phloem. Here you cannot see any cambium in monocots. And hence no secondary growth, because cambium is the meristematic tissues which give rise to the secondary xylem and phloem. If cambium is absent in monocots, there will not be any secondary growth. These vascular bundles are called closed vascular bundle. There present the protoxylem and metaxylem. What are they? The protoxylem is the first formed xylem, which is the primary xylem, and metaxylem is later formed part of the primary xylem that develops after the protoxylem. If xylem and phloem are arranged alternatively along different radii in a vascular bundle. They are called radial vascular bundle. Here you can see this one is phloem and this one is xylem. They are arranged in different radii. Hence they are known as radial vascular bundle. This arrangement can be seen in roots. If xylem and phloem are jointly situated along the same radius, those vascular bundles are called conjoint type. These are the conjoint type vascular bundles. Here phloem is located on the outer side of the xylem. Xylem towards inner side and phloem towards outside. It is seen in stems and leaves. In radial type, the xylem and phloem are radially arranged in different radii. And in conjoint type, they are joined together in the same radius. Now moving on to the ground tissue system. Tissues except epidermal and vascular tissues, they are known as the ground tissues. It consists of parenchyma tissue, collenchyma tissue and sclerenchyma tissue. Parenchyma cells, they are living cells, they are thin-walled and unspecialized, but they can perform a wide variety of functions depending on where they are located in the plant. They are present in the cortex, pericycle, pith, medullary rays, in primary roots and stem. Don't worry about these new terms. We will learn all these terms in this video. Collenchyma provides flexible support and allowing the plant to grow and bend. This is the reason the plants can bend because of the collenchyma tissue. They are present in young leaves and shoots. Next is the sclerenchyma tissue. It is a type of supporting tissue in plants that provides rigid mechanical strength. Parenchyma and collenchyma are living cells, but sclerenchyma cells are dead at maturity and they have thick lignified cell walls. It can be seen in mature stem and bark. In leaves, the ground tissue consists of thin-walled chloroplast containing cells called mesophyll cells which help in photosynthesis. We learned about the three types of tissue system: epidermal tissue system, vascular tissue system and ground tissue system. Now let's learn about the anatomy of monocots and dicots. First, let's learn about the anatomy of dicot roots. We know that dicots are plants who has two cotyledons on its seeds. Example, mango, tomato and sunflower. The outermost layer is known as the epiblemma, the epidermal layer. Here, this is known as the epiblemma. Here we can see the root hairs. Many cells of the epiblemma protrude out in the form of root hairs. The area beneath the epiblemma, this dark area is epiblemma, and beneath this area, this entire thing is known as the cortex. The cortex has many layers of cells, you can see, and these cells are parenchymatous in nature. We know that parenchyma cells are storage in nature. They have intercellular spaces in between them. Here you can see a lot of intercellular spaces are between the parenchymatous cells of the cortex, which help in gas exchange and storage. The inner layer of the cortex is known as endodermis. Here the outer brown layer, this is known as endodermis. That is the inner layer of the cortex. It is endodermis. It has single layer of barrel shaped cells. You can see the structure of cells here. They are barrel shaped cells. The radial and transverse walls of the endodermal cells have presence of suberin deposition. Here you can see suberin deposition will be like this in the radial walls. What is suberin? Suberin is a water impermeable substance and they are known as Casparian strips. What is that? Casparian strip. The radial walls of the endodermal cells have the presence of water impermeable suberin deposition. They are called Casparian strips. They regulate the water and nutrient movement between the cortex and the vascular tissue. This is the vascular tissue, which is the xylem and phloem, and this is the cortex. And these Casparian strips, they regulate the movement of water and nutrients between the cortex and vascular tissue. Next to the endodermis, there are thick-walled parenchymatous cells, which are known as the pericycle, the inner wall. This one. This is known as the pericycle. Pericycle. This is the pericycle. It gives rise to new root apical meristems for lateral roots, which results in secondary growth. And it also gives rise to the vascular cambium. We learned about cambium, right? That gives rise to secondary xylem and phloem. Let me draw the cambium as well. So this is the cambium. The central part of the root is known as the pith, which contains the parenchymatous cells that help in storage of nutrients and oil. Parenchymatous cells are present in between the xylem and phloem, which give rise to the support. These are the parenchymatous cells in between the xylem and phloem. They are known as conjunctive tissue. Here, this is the conjunctive tissue. The tissues between the xylem and phloem. These parenchymatous tissues are known as conjunctive tissue. There are 2 to 4 xylem and phloem patches. This is 1, 2, 3 and 4. Here 4 xylem and phloem patches are there. The tissues inside the endodermis, that is the pericycle. Here this is the pericycle. The vascular bundles, that is the xylem and phloem, and the pith. Together they are known as stele. They are known as stele. That is the pericycle, this pericycle, the vascular bundles and the pith. Together they are known as stele. Now let's learn about the monocotyledons roots. Most of the structures in monocot is similar to that of dicot. However, some variations are there. Monocots have a greater number of xylem and phloem bundles. Here only 4 are there. Here 6 to 7 are there. Xylem and phloem bundles, 6 to 7 are there. Pith in the central portion, which is large and well developed, and there is no cambium present in between the xylem and phloem, and no secondary growth happens in monocots. Cambium is present in dicot root, but it is absent in monocot roots. Next, let's learn about the anatomy of stem in dicot plants. The outermost layer of stem is known as the epidermis, which is covered with a waxy layer known as the cuticle, and the stem may have trichome, which is the epidermal hair. The area beneath the epidermis, this area is known as cortex, and cortex has multiple layers of cells. The outer layer, this brown layer, is known as the hypodermis. These cells are the cortical cells, and this brown layer is known as the endodermis. Hypodermis has collenchyma cells, which provides the mechanical support in young stems. And this cortical layer cells can be found below the hypodermis. It has parenchymatous cells with a lot of intercellular spaces. The inner layer of cortex is known as endodermis. It is the innermost layer of cortex which are rich in starch grains. Hence it is known as starch sheath. So the area below epidermis is known as cortex. Cortex has 3 different areas. This is hypodermis, cortical layers and endodermis. Endodermis is rich in starch grains, hence known as starch sheath. Similar to root, the cells inner to the cortex is known as the pericycle. Here, this is the pericycle. Inner to the endodermis is known as the pericycle. It is present above the phloem. So this is phloem and this is xylem. It has sclerenchyma cells which are thick-walled, lignified and dead cells. These cells provide vital mechanical and protective functions. In between the vascular bundles, there present a few radially placed parenchyma cells. In between the bundles, there are here radially placed cells are present. They are known as the medullary rays. They transport water, minerals and food between the vascular bundles. These medullary rays, they are radially placed cells. They transport water and food in between the vascular bundles and from vascular tissue to the cortex and pith. In between the xylem and phloem, there present the cambium. In between xylem and phloem, here cambium will be there. This one. And let me draw with another color. This one. This is the cambium between the xylem and phloem. We learned protoxylem and metaxylem earlier in this video. Protoxylem is the first formed xylem. This is the first formed xylem. Metaxylem is the later formed one after the protoxylem. In dicot stem, the protoxylem is located towards the center, that is towards the pith, and metaxylem is towards the periphery. This arrangement is known as endark. This is known as endark arrangement. That is the protoxylem towards the pith and metaxylem towards the periphery. This arrangement is known as endark. And the central portion of the pith has many parenchymatous cells in it. Now let's learn about the anatomy of monocot stem. Similar to dicot stem, monocot stem also contains epidermis. This is epidermis. Below the epidermis, hypodermis is present. This is hypodermis. In dicot, the epidermis has collenchymatous tissue. But in monocot, this hypodermis consists of sclerenchymatous cells. In dicots, they contains collenchyma cells. But in monocots, it contains sclerenchyma cells. The vascular bundles are not in ring form. The vascular bundles, that is the xylem and phloem, in dicot, they are arranged in a ring form. In dicot stem, the vascular bundles are arranged in ring form. It is a characteristic feature of dicot stem. That means if you can see ring arrangement of vascular bundles in a section of stem under the microscope, we can confirm that it is a dicot stem. But in monocots, the ring arrangement of vascular bundles is not seen. Instead, they are scattered. Each vascular bundle is surrounded by a sclerenchymatous sheath. Each vascular bundles in monocot is covered by a sclerenchymatous sheath. Unlike the dicot stem, monocot stems do not have distinct cortex, endodermis, pericycle or pith. Instead, there is a presence of large parenchymatous ground tissue. There is no differentiation like pericycle and pith, but there is a group of parenchymatous tissues are there. Vascular bundles are conjoined as xylem and phloem are present in a singular radii, but the cambium is absent and hence called closed vascular bundle. Peripheral vascular bundles, they are small. Here they are towards the periphery. These vascular bundles are small, and towards the inner side, these vascular bundles are comparatively big. Phloem parenchyma is absent. Water containing cavities are present within the vascular bundle. Now moving on to the dicot leaf, which is also known as dorsiventral leaf. A dicot leaf is called dorsiventral because it has distinct upper and lower epidermis. The upper epidermis is known as adaxial and the lower epidermis is known as abaxial. The vertical section through the lamina of leaf shows three distinctive parts: the epidermis and the mesophyll cells and the vascular system, that is the xylem and phloem. Which all are the three areas? Epidermis, mesophyll cells and vascular system. The outer layer of leaf is called epidermis and it covers both the upper and lower surfaces. The upper surface is covered by adaxial epidermis and lower surface is covered by abaxial epidermis. Epidermis is covered by a layer of cuticle. This dark green layer you can see, that is the cuticle. The abaxial, that is the lower epidermis, has a greater number of stomata than the adaxial. Here in adaxial side there is no stomata at all, but here in abaxial side you can see many stomata. The lower surface is less exposed to direct sunlight, so transpiration is lower. If that many numbers of stomata are present in the adaxial epidermis, water loss will be more because they are exposed to direct sunlight. The tissue between the adaxial and the abaxial epidermis is called mesophyll. Parenchyma cells are present in the mesophyll which bears chloroplast. Hence, photosynthesis is happening in the mesophyll part. The parenchyma present in the mesophyll can be of two types. They are palisade parenchyma and spongy parenchyma. Palisade parenchyma is present near to the upper adaxial epidermis. It is the main site of photosynthesis. Due to the presence of large number of chloroplasts, the cells are elongated. You can see here the cells are elongated and are arranged vertically and parallel to each other. The intercellular spaces between them are very less. The next kind of parenchyma cells in mesophyll area is known as spongy parenchyma. They are present beneath the palisade parenchyma above the abaxial side. They have large intercellular spaces. You can see here. Large intercellular spaces are there between the cells. The large intercellular spaces allow free movement of carbon dioxide and oxygen and water vapor essential for photosynthesis and respiration. Vascular system, that includes the vascular bundles which are present in the veins and midrib. The vascular bundles are present in the veins and midrib of leaves. Vascular bundle are surrounded by a layer of thick-walled bundle sheath cell. These are called the bundle sheath cell. The vascular bundle is covered with thick-walled bundle sheath cells. These bundle sheath cells form a protective layer around xylem and phloem, shielding them from damage. They also control the movement of substances like water, nutrients and sugars into and out of the vascular tissue. In C4 plants, Calvin cycle takes place in the bundle sheath cells, which you will learn in chapter 11. I have provided the link for that chapter in the description box. Please visit. Now let's learn about the anatomy of monocot leaf, which is also known as isobilateral leaf. A monocot leaf is called isobilateral because both the upper and lower surfaces are structurally similar. There is no abaxial and adaxial. They are similar. They appear almost the same in terms of anatomy and function. That is why they are known as isobilateral leaves. Most of the features of monocot leaves are similar to dicots. However, there are some differences. Stomata are present in both the surfaces, and mesophyll is not differentiated into palisade and spongy parenchyma. The grass leaves. Some cells of adaxial epidermis modify themselves into pulliform cells. So here imagine this is the adaxial side of the grass. This is a bulliform cell. Okay, this is known as bulliform cells. Their function is to open and fold the leaves. When the plant has adequate water, bulliform cells are turgid and the leaf remains open for photosynthesis. Turgor pressure is the pressure of water inside the cell pushing against the cell wall, right? So during water stress or drought, bulliform cells lose turgor pressure, becomes flaccid, that means they become soft and wetted, and cause the leaf to fold or roll inwards. This reduces the exposed to surface area, minimizing water loss through transpiration. That's it about the chapter Anatomy of Higher Plants. If you like this video, please share it with your friends. If you have any queries, put it in the comment box. Thank you.